Hello and welcome to KAUST Live. This week we have a special treat for you. The CBRC is having a Big Data Analyses on Evolutionary Biology conference, and so we've asked some of the researchers to come and speak with us about the keynotes that they're giving, some of the research they're doing, and what's going on with them. So we have Kelso and Sudhir Kumar with us today. They've both given keynotes earlier in the day at the conference. So thank you for joining us. I really appreciate it.
Thank you.
Thank you.
Yeah. So I wanted to ask you both a little bit just to intro who you are, your institution, and a little bit of the work you do. So maybe we can start with you, Janet.
So my name's Janet Kelso, and I'm a researcher at the Max-Planck Institute for Evolutionary Anthropology, which is in Leipzig in Germany. And for some years now, we've been working on generating the sequences of ancient humans, Neanderthals and other human forms. And my part of that is really the computational part. So I'm a computational biologist, a bioinformaticist. And what we've been doing is figuring out how to analyze the sequences that we get from these old bones, and then trying to learn something about both Neanderthals, Denisovans, this other group that's related to Neanderthals, and also something about ourselves, so how we as modern humans are related to the Neanderthals and how we might have interacted with them and what we've gotten from them, so to say.
Very good. Very interesting. And we'll come back to that. So Sudhir?
So I'm at Temple University. I direct an institute for Genomics and Evolutionary Medicine. And as Janet said, a lot of researchers, and including her own group that she works with, are generating lots amount of data about human data, data on disease in humans, data on other species, because we are able to now see what other species DNA looks like, and we are able to do it for a large number of species that are, that are close to humans with respect to their similarity to us, and the species that are very different from us, species that are pathogens, that give us diseases, others that make us healthy. So all of this data, tremendous amount of it, is what we call big data, because we have huge genomes and large number of DNA sequences. So what my group does is, it does comparative analysis. It takes DNA from different humans, different individuals, different species, different pathogens, and compares it to each other, and then build methods and tools that actually allow researchers like Janet and many others to make sense of what it is it's telling us. Do we have certain things we want to test? Do we want to know where did the flu strain that is all around us today came from? What is the origin of HIV? So we can ask all these amazing questions from big data. So what my group does is develop methods for it, along with many other people are doing that, and also develop actual software tools to analyze that data.
So Sudhir, one of the things you're perhaps best known for is MEGA. Talk a little bit about that and the paper that's earned you a bit of notoriety.
So MEGA, as the name suggests, is something that turned out to be big. When we first did it, it was relatively modest effort. I began writing the software MEGA when I was a graduate student, and we have been developing it since then. It's been 24 years actually working in this project. And the idea behind MEGA was to make a tool that almost any student and also any researcher could use for analyzing DNA sequence data. And the data can come from pathogens like bacteria, virus. It could actually come from different humans, individuals in a population, in a group. And essentially when you use this tool and the algorithm it has, many of them are published by us and many others by others, you can start to make sense of your data. So you can see if you take the data from a viral strain or flu, and you can compare the sequence of flu over time from different populations, different regions of the world, you can start to tell, oh, this particular flu strain came from Hong Kong or China or India or any other place. You can start to place that, and that's, as you know, is very important to know how epidemics travel. So these kinds of tools allow people to use their data effectively. And the tool is, the biggest thing about this tool is very user-friendly. So you can actually do these analyses in a way that you can do on your desktop, a large-scale analyses, and then make inferences. And these are the inferences you see in published literature as well as newspapers about when did HIV arise, when did the patient O existed in America, for example, for HIV, when did a certain bacterial strain come in our population, or how similar are two genes in humans? Same kind of question is there. So you can answer those questions with that tool. And as over time, more and more people, more and more researchers are using DNA as a tool to understand biology, this tool Mega has become more and more useful.
Right.
And so now used for classroom teaching in high schools, in colleges, and of course research laboratories.
And you had said before we started the interview that the paper's pretty highly cited. Where is it in the world of cited papers?
So the Mega software papers are extremely well cited. So one of the papers in top 100 of all times, and the other one is like top 150 of all time. And overall, Mega has been cited more than 100,000 times.
Right.
But that simply is because people have a need,
Right.
and the software is fulfilling the need. When we develop these software, we develop them primarily as tools because we can, and we want to give back to the community by developing methods and tools that are useful. But sometimes you get lucky and tools become widely used. And this is one of those cases, and I'm happy to have that.
Right, right. Very good. So, Janet, you were talking about ancient genomes, and I'm wondering about some of the analyses that Sudhir was talking about. Tell us a little bit about analyzing ancient genomes and how that even came about for you.
Sure. So until the mid-2000s, until around 2008, it wasn't really possible to analyze the nuclear genomes of ancient humans. And this is simply a feature of the fact that the DNA is so poorly preserved. So when you extract DNA from an ancient bone, it's fragmented into really tiny pieces, it's very badly chemically damaged, and it sits in this huge sea of microbial sequences. So it's not like taking DNA from you or taking blood from you and pulling DNA out of it, where I get your DNA and it's pretty clean and I can sequence it. What you get is this whole mess of kind of microbial sequences, environmental sequences, and hidden in there somewhere, the sequences from the individual that the bone belonged to.
Right.
And so the very first challenge when we started sequencing Neanderthal bones, nuclear DNA from Neanderthal bones in 2008, was to figure out how to fish out the sequences. And what we did is we used the human reference genome, so the genome that was sequenced, the very first human genome that was sequenced as a reference, as the bait, if you want to put it that way. We used it to pull out sequences that are similar, because we know Neanderthals and modern humans, as Sudhir has said, are rather similar to one another. And so because the sequences are rather similar, you can kind of use an alignment tool, which is something else that Sudhir has talked about developing, to pull out sequences that are Neanderthal or potentially from Neanderthals. And so the very first approaches were, sort of using what we call a reference-based method, where we have human reference and we use that to find Neanderthal sequences. And along with that comes all sorts of other challenges. How do you identify contamination from people today who've handled the bone?
Right.
And we've been developing methods for finding contamination. How can we be sure that we have authentic ancient DNA? We know that the Neanderthal DNA or old DNA carries a very particular pattern, chemical damage pattern, and we use that as kind of a signal that there's ancient DNA present in our samples. And so my group has been involved in building tools for processing sequence data from ancient genomes, looking for contamination, figuring out how to do the alignment, and then moving downstream, working with many other groups on how we can understand how we're related to the Neanderthals. And for a long time that wasn't totally clear.
Yeah.
Now we know that the Neanderthals are a sister group to us, if you want to call them that. We share a common ancestor around 600,000 years ago. But they're not our direct ancestors, right? We're kind of cousins.
Right.
And the Neanderthals, they lived in Europe and Western Asia, and modern humans, we know from genetic history, from anthropology, that they arise in Africa. And then they move out across the world, kind of taking over as we know it today, right, most of the planet. And I think one of the standing questions had been, when we did that, was there any interaction between Neanderthals and modern humans?
Right.
Did we interbreed? What happened to the offspring? And when we finally had a Neanderthal genome, we could answer that question for the first time. And we could say, yes, actually, everyone who lives outside Africa today has a tiny bit of Neanderthal DNA in them. Around 2%, if you're European or Asian, of your genome comes from interbreeding in the past between your ancestors and the Neanderthals.
Right.
And that's totally cool, right? We can now say, well, there's a little piece of your genome that's Neanderthal-like.
Right.
And slowly we're starting to say, well, what does that do? Does it have any effect on people today?
Right. This is a fairly recent finding, I believe.
Yeah, so the discovery that there is around 2% Neanderthal DNA in people today was made in 2010, I think it is. So it's in, but this idea, these ideas that are developing about what effects that might have, this is really recent work. It's in the last year or two that people have started to get a handle on where in the genome are these fragments of Neanderthal DNA and what kinds of genes are they affecting. And what we're starting to see is, it's lots of things to do with environmental adaptation. So lots of things to do with how we, or what we borrowed from the Neanderthals, if you want to put it that way, were adaptive variants, things that allowed us to adapt to the climate, to the foods, the pathogens that were present. So we've kind of borrowed all these things that the Neanderthals had developed over many, many, many thousands of years while they lived in Europe and Western Asia. And we kind of got a bit of a head start on that by breeding with them and borrowing some of those variants.
Right. So does part of this involve, understanding the distance between different genomes? And I wondered, Sudhir, if you could talk a little bit about, as this is part of your experience.
Yeah, so I think, as Janet mentioned, you are comparing Neanderthal DNA to human DNA.
Yes.
And we compared humans to other humans, the living humans or modern humans. And so essentially when we compare two DNA molecules, one from one person and one from another one, you can see some differences.
Right.
And these differences, the number of differences is basically a measure of evolutionary distance.
Right.
Between two brothers or two sisters or brother and sister, this is very small, almost negligible. And between Neanderthals and humans, for example, modern humans, it is larger. And comparing humans to chimpanzees or gorillas or other species is much, much larger compared to that. Right. So essentially, evolutionary distance is very simply the amount of change that has happened between two DNA sequences.
Right.
And depending on where they come from,
so the question that raises for me is, you're talking about ancient DNA in particular that's fairly fragmented, which seems like a crazy puzzle in and of itself, and then you add these evolutionary distances, which I'm sure it's not just one species to one species. I'm sure it's a bit more complicated at times. So talk a little bit about how you work all of that out. I'm headed for the big data here. How does that help? How does that help?
I hesitate to say we have big data. With it's a lot of sequences, but with the Neanderthals that we have a rather limited number of samples where we can actually even get DNA. So we're talking about lots of sequences, but from, at this point, still a rather small number of individuals, individual Neanderthals.
Right.
So we have genomes from 2 Neanderthals.
Wow, just 2?
Just 2.
Okay.
Good genomes, where we would say we know very confidently at each position in the genome what nucleotides, what bases that Neanderthal individual carried. So those genomes, I would say, they're as good as the genomes of people that we would sequence today.
Wow. Okay.
We have a couple more. We have 5 or 6 more genomes where we have much lower coverage and we're much less confident about exactly the bases in those Neanderthals, but it gives us more information about the Neanderthal populations. And so what we're interested in is trying to understand, how did Neanderthal populations themselves change over time? Right. These Neanderthals span a period of 100 to 150,000 years. And the 5 new Neanderthals that we've just been working on, they are what we call late Neanderthals. They're Neanderthals who were living at the very end of the period, just before Neanderthals disappear, which is around 40,000 years ago. And those populations look, how were they related to the earlier Neanderthals? And was there any evidence that there was also gene flow into them from the modern humans that they met? Interesting,
yeah. Is this, would you say, a body of the research that's led to your recognition by L'Oreal? I know that we have a professor on campus who also was recognized by L'Oreal. So talk a little bit about that experience.
So actually it's not, because the L'Oreal award that I received was in 2004.
Wow,
it was prior to moving to Leipzig to work on these Neanderthal genomes. It was actually a fellowship in recognition of my PhD work, which was on a completely different topic.
Okay, very good.
It was computational work on understanding gene expression.
Well, talk a little bit about that experience as a young scientist then.
Sure.
I mean, you were...
I was, of course, extremely honored. I was pretty surprised, actually. Very honored to be considered. and then finally to be awarded the award. Looking back, it's many years ago by now, and looking back, it's funny to think that at that point in time we had no human reference genome. So I was working on problems trying to work out how many human genes are there,
Right.
where are they expressed in the body, using data from ESTs, these short cDNA tags.
We used to have a lot of genes then.
We had a lot of genes when we had ESTs. So there was a lot of work going on in the early days before there was a human reference genome to try and figure out how many genes do humans have. And many people kind of speculated that humans are so complex that they must have many more genes than flies and worms.
Right.
That turned out not to be true. We have about the same number of genes, or maybe slightly more than flies and worms. But this was a big project in the lab that I worked in, kind of calculating human gene numbers from ESTs. And my work was specifically on trying to understand gene expression and tissue-specific gene expression, and building controlled vocabularies for describing where and when genes are expressed during development and also anatomically.
Yeah. So that gets to how rapid the change in the way that the research has been done in the industry, right? I mean, that's only 14 or so years ago.
That's
So
true.
now we're to the place, and Sudhir, you can talk a little bit about this, like where entire viral genomes are mapped very quickly, as you said, and compared to thousands of other samples.
That's
to
right.
Get history of a virus. So talk about doing that.
Yeah, precisely. So basically, we have reached a time where you can sequence a complete virus in few hours.
Right.
So you have an epidemic, and you have a lot of people in the population, and you can actually get data on a matter of days
Right.
from large number of individuals, a large number of strains. Right. In the case of HIV, some people have large number of strains because HIV within a person constantly changes, mutates very, very quickly, and makes its own populations.
Right.
And sequencing those strains or sequencing strains all over in a population has become very easy. So ultimately what that has meant is this big data revolution, which is data deluge. Every day you have a huge number of new sequences, long DNA sequences being deposited in the gene bank. Or people are able to deposit them in different places, different repositories. And then there are researchers like Janet and I and others who in fact go to these repositories and download the data. So this is the era of new biology. This is a biology in which I don't have to, in my own laboratory, have the organism that I study. I am able to get the data directly from this bank or a collection of banks, and then I'm able to contribute to the knowledge by analyzing them in my own way. So in a sense, the science is becoming more and more democratic. Anybody can go after these data, and we have a mandate to make the data available to everybody as necessary. So this means a lot more people are putting their mind to it. And it's sort of a crowdsourcing by people who are a crowd of individuals who can do bioinformatics, who can do computational analysis. And that's where I think people need better tools, like some we develop and some are developed by Janet and others. And they need the data in a form that is informative. So there's a lot of information telling where the genes are in a big genome or DNA sequence. This is a big challenge. You constantly discover that these viruses or these bacteria interchange genes. So it's not like all the bacteria have the same set of genes that they've acquired through their predecessor bacteria. They exchange genes too. And that I think is a very exciting area, which has implications for antibiotic resistance, drug resistance, all kinds of resistance, also in becoming more infectious.
Right.
It's another problem. So I think the DNA sequence analysis, comparative genomics, bioinformatics has now become center place in biological laboratories.
It's kind of hard to underestimate how much technology has changed this field of human molecular evolution, or even just molecular evolution in the last not even 10 years.
Yeah.
It's a fundamental difference to where we stood 10 years ago in terms of the amount we understand, or the amount of information we have, maybe that we don't completely yet understand, but the amount of information we have to mine about, I'll talk about what I know about human variation, for example. We have the genomes of thousands of people. We have full medical records for many people, and we have also much self-reported information about those people that are incredibly valuable ways to learn about the origin of disease, susceptibility to different diseases,
Right.
And also about human evolution, about how humans have changed over time to adapt to their to adapt to new environments, how the changes in behavior, for example, farming, have modified or have had effects on our genomes.
Right.
It's really, and underlying this, I think we haven't said this really explicitly, it's sequencing technologies. The ability to sequence genomes really rapidly is something that started in 2008, around that time. It's really, really recent. And so these things tools are still being developed, and people are still getting to grips with what's possible.
Yeah, so imagine, you can imagine for people who are not doing sequencing, it's like rise of internet. How it has revolutionized your buying, right? So think about how everything is changed.
That's right.
The internet has been disruptive technology for everything. I think sequencing, in a sense, has been disruptive technology to many questions in ecology, evolution, disease. Now, the first thing we think about is, can I somehow use DNA sequence technology to quantify an ecosystem?
Yeah.
You take a soil sample, you sequence the soil sample, and you can tell what is the community that is living in this sample. You do the same exact thing in a human gut, or human face, or any other part of humans, and you can quickly see the difference.
Yeah.
And that's all brought upon by the technology, which
Technologies.
is sequencing technology.
Right.
So I often say that genome is no longer, we are not doing genome analysis for genome's sake.
Yeah.
We are now using genome or DNA as
It's a molecular barcode.
like a marker
yeah.
for everything.
Right.
Like we do now for crime scene investigation. Same thing. Instead of fingerprints, now you are actually doing DNA fingerprinting.
Right.
So what has happened is the DNA is now, its applications are everywhere. And behind all those applications is one thing, comparative analysis of DNA from different people and different species. And I think that's why this area, this big data analyses in evolutionary biology is important. It's not just this old armchair evolutionary biology idea. It's actually everyday practical analysis, paternity analysis, crime scene investigation analysis, family cohort studies, finding genes that link with diseases, finding our most recent cousins,
Right.
essentially going on to building the tree of life.
Right.
And we
You
can
can do the same,
Yeah,
right?
you can really start to understand, I mean, for example, the field of human evolution, of archaeology, that uses fossils to understand human history. It's been really important in understanding modern human history, but also archaic humans. But DNA gives you an amazing tool set to really understand, if I find a bone, what species is it from? Is it from a horse? Is it from a bear? Sometimes you can tell that by morphology. You can look at the bone and say, oh, this is very likely from a bear. But sometimes you can't because you have a tiny little fragment. And a good example is a new human group called the Denisovans that were discovered by DNA sequence alone. So there was a small bone. It was thought to perhaps be from a modern human, maybe a Neanderthal. But we sequenced it. We saw that it wasn't a Neanderthal. It also wasn't a modern human. And it was this whole new group that had never been identified just on morphology, and we don't know why that's true, but DNA discovered a new group, a new human group.
Right. So what does this bring us to 10 years down the road then? Is this a custom-made healthcare? Maybe you can both give a little idea in your subfields for how that's going to play out, you think?
That's a great question. I think we're still looking, kind of trying to look over the horizon. I don't think we know yet where this will bring us. I think there's a lot of hope for personalized medicine, for personalized healthcare, and Sudhir talked a little bit about that today.
Is that phylomedicine? Is that what that's referred to?
So
essentially?
phylomedicine is more to do with how we use this long-term history of humans and their ancestors, whether they are shared with other mammals or other backboned animals or other invertebrates. How can we leverage that to understand human DNA and its changes? Because we can't experiment on humans, obviously, and the human genome is vast, so you can't actually do experiment in the laboratory all the time. So what you need is a prediction method, forecasting. And phylomedicine is to use a long-term history knowledge to forecast about short-term, which is, if I see a mutation in my child, what does it mean?
Right.
And so that's what science of predictive medicine is, and that's where phylomedicine is one way of doing it. It is in addition to many other analyses that people do. But what I see down the line in 10 years, I feel like is one possibility is that we will have essentially a genome from everybody who's around. Maybe 5 billion people will donate their DNA sequence for genome sequencing. And they will also tell us all their behaviors, idiosyncrasies, niceties, or whatever it is, and diseases. and then imagine having a Google where a person can just take their DNA, look at every place where they have what we call a mutation, and just search and say, hey, who else has this mutation? And then look at what these people are like, right? So that would be a cool thing. So I think what I envision in 10 years from now is really human Google. So you Google, you know. So this is what I think will happen in 10 years, at least from the perspective of simple, because it doesn't really require any inference. What you're looking for is
it
similarity.
sort of exists in some places already. It's sort of mini hubs.
So it's starting to,
personalized genome testing.
Personalized genome testing. When you do, you go to a sequencing
company.
place like Ancestry or 23andMe or whatever. There's so many places.
Yeah.
You get the DNA profile, and they tell you which particular parts of the defects or mutations you are carrying.
Right.
What are they associated with?
Right.
The often problem is we know associations, we don't know causation. So that's why they remain within the realm of it's good to know, but actionable they are much less than ideal. So you want to know, but at this point we don't know a lot of things to what we can do.
Right.
But these information are actually being used in oncology all the time, in HIV cocktail selection all the time. So I think we are reaching the next generation of medicine where we'll be better informed by our own DNA. So I think that's what I see 10 years from now. It will be much better.
Very good. Well, I think we'll leave it there. We've gone from Neanderthal genome to human Google. I don't think we're going to beat that at this point. And thank you for joining us. Tomorrow we're going to be speaking with Shozo Yokoyama. He has a keynote tomorrow as well, I believe. And that's all for now. Thank you.